We extracted 5 randomly spaced soil cores using a soil core sampler and homogenized them into a collection bag to avoid any potential sampling bias resulting from the presence of potential microhabitats within any given plot. The collection bags were opened and placed into a fume hood at room temperature in the lab to desiccate completely. Here's how that looks. This triggers cryptobiosis within the microbial population and allows for more efficient preservation and handling. We then measured 10 grams of soil and transferred it into a petri dish. We then crushed the remaining granules of soil that were large, as you can see on the left afterwards. Once ready for observation, we then watered the petri dish to 100% saturation plus 2 milliliters of excess water. Laboratory stimulated existment was conducted via the resaturation of the soil, and up to 14 days were allocated to allow all organisms to exist, accounting for varying reemergence rates. The following process took place on each observation day, which was day 2, 4, 7, 11, and 14, to collect a representative population survey. As pictured here, the petri dish was tilted at 45 degrees, allowing the excess to gather at the bottom against the petri lip as runoff liquid. 0.5 milliliters of this runoff was extracted via micropipette and placed into an Eppendorf tube for the next step. The Eppendorf tube was then allowed to settle, or centrifuge, which forms precipitate of soil and liquid supernatants. For each slide, we sampled 50 microliters from immediately above the precipitate along that dotted line to collect the organisms that settle on the solid detritus. To create the slide, the sediment should first settle at the bottom of the micropipette tip, and the researcher then gently touches the meniscus to the slide in small, separated dots, lifting after the slide makes contact. The slide pulls the liquid down via surface tension, accompanied by denser concentrations of soil. This creates little soil islands, as you can see on the left, which equally separate the total soil concentration, as opposed to the usual method of just bunching it all into the middle, like the slide on the right. Our devised method on the left is beneficial because decreasing the amount of space in which an organism can hide yields clearer observation opportunities, especially with completely transparent zones between islands for the clearest possible contrast. Let's take a look under the microscope for an example of this benefit. Within this red circle is an organism that is hardly distinct from the environment around it. This is because the soil is so densely concentrated into one area that it makes it difficult to distinctly identify a transparent organism, even if it's moving. This was an example of the rightmost slide preparation. Let's take a look at our method on the left. Here is that same species, but captured using our method of sample preparation. As you can see, our method retains a clear benefit of increasing contrast and transparent medium between soil islands for the best possible chances of quantification. In order to best record our data, we increase the number of slides and decrease the soil concentration on each in order to minimize the amount of microorganisms to best suit our counting method. We scan the slide in a grid-like fashion, going slowly and counting every organism that we came across. If there are too many organisms to count accurately, or if they're going too fast and swimming between grid cells while counting, the slide is reset and we start over. Once the slide is accurately quantified, we clear it and begin a new slide, using the same method as before, taking from the dotted line of the Eppendorf tube. This is done until the supernatant completely runs out,